Hey everyone, welcome back to our USMLE Step 1 crash course. It's me, Dr. Deepak. Today we are diving into crucial concept from neurological embryology, how the early nervous system forms starting from the notochord and progressing to the neural plate, neural tube and neural growth cells. In the same time, we shall also break down all our plate versus basal plates and give you a key mnemonics. So let's start in. First of all, we need to know about gastrulations. As you know, there are three germinal layers: ectoderm, mesoderm and endoderm. Let's draw it. Ectoderm, mesoderm and endoderm. So here is the role of notochord. It's very important for us. The notochord actually is a rod-like structure derived from mesoderm in the midline of embryo. And its main job is to induce the overlying ectoderm to become neuroectoderm. This is a very important concept here. So notochord is a rod-like structure derived from mesoderm in the midline of embryo, and its main job is that to induce the overlying ectoderm to become neuroectoderm. So what is neuroectoderm? Neuroectoderm is a specialized tissue that form neural plate. So here is the important point. And in a clinical connection, here is a point. In a clinical connection, notochord become nucleus pulposus of intervertebral disc in adults. What is this nucleus pulposus of intervertebral disc? It is a jelly-like center that can herniate in disc prolapse. So this is the clinical connections of notochord. Okay, you need to understand in adults, the notochord regresses and its remnant become nucleus pulposus of intervertebral disc, and ultimately that can herniate in disc prolapse. So important now. Another main point is that neural plate. So neural plate is a flat sheet. Here is the flat sheets of neuroectoderm that forms above the notochord. Around the 3rd to 4th week of development, the edge of this plate, the edges of this plate elevate to form neural folds, which they infuse in the midline and creating the neural tube. At first, after neural plate, they make the neural folds. Okay, neural folds. And these neural folds, after neural folds, what will happen? After these neural folds, so these fuse in the midline, creating the neural tube. After that, they make a neural tube. So this is important. The neural tube eventually they will give rise to CNS. They will give rise into CNS. To the CNS means brain and spinal cord. But wait, there is something very interesting. Magical things is also happen at the tip of the neural folds. At the tip of the neural fold, that cells at the crest separate and migrate away. So migrate away. These cells are called as these are called as a neural crest cells, NCC, which give rise to wide variety of structures. Here is the mnemonics, and the mnemonic is magic. As you see here, magic. M for melanocytes, adrenal medulla, ganglia, iris stroma, craniofacial structures. So clinical tips are here is that if neural tube closure fails, we get neural tube defect. We will get neural tube defects. For example, Spina bifida occulta and anencephaly. So now most important part is that now the neural tube is formed, but its lateral wall differentiates into two key regions. That is alar plate, which is dorsal, becomes sensory neurons, and the basal plate, which is ventral, becomes motor neuron. This division follows a gradient of molecular signals. The first one is BMP, bone morphogenetic proteins from the overlying ectoderm induce the alar plate induced, and that is sensory neurons, and SHH, sonic hedgehog, which is secreted by the notochord and floor plate, induces the basal plate, and that is the motor neuron. So here is the mnemonics: SSS in a mnemonic, BMP on the back. It means that BMP back sensory, SSS in the soil it means SSS from the below, the motor. So think of this like a spinal cord, Think of this like a spinal cord organization. Here is a spinal cord, there is a central canal, and here is, dorsal horn. Dorsal horn, that is sensory, and this is a ventral horn. This is a motor. Okay. The same orientation is maintained from embryology into adulthood. So let's review the main key points here. So the first is here notochord. Actually, notochord induces neuroectoderm and neural plate. Neural plate folds into neural tube and neural crest cells. Neural tube, of course, ultimately becomes CNS, and neural crest becomes PNS and other tissue. Fourth point is alar plate, actually dorsal horn or dorsal side, and it is a sensory neuron. It is a sensory neuron and induced by BMP. Basal plate is a ventral and motor neuron, and it is induced by SSS. For the step 1 tips, the most important questions in steps 1, neural crest derivatives are the favorite exam topic. Know the signaling molecules, especially BMP for sensory, SSS for motor. Remember, the notochord becomes the nucleus pulposus, not a part of nervous system. Thank you very much. Dear friends, in our last video we talked about how the neural plate folds into neural tube and gives rise into CNS. Today we shall zoom in on what happens after neural tube forms, how it regionalizes into different brain vesicles, and how it gives rise to structure of CNS and PNS. This is very high yield step 1 materials. So get ready to take note and remember the mnemonics. So first of all, let's talk about the primary brain vesicles. At week 4 of embryogenesis, the closed neural tube begins to expand and differentiate at its rostral. Its rostral head end, this results into 3 bulges or primary brain vesicles. The forebrain is also known as a prosencephalon. Midbrain is also known as a mesencephalon. And hindbrain is also known as a rhombencephalon. And here is the interesting mnemonics to remember it. It's very easy to remember. PMR, primary medical reference. P for prosencephalon, M for mesencephalon, R for rhombencephalon. So by week 5th, these 3 vesicles further divided into 5 secondary vesicles. The prosencephalon, the forebrain, divided into telencephalon and diencephalon, while as mesencephalon stay as a mesencephalon. And rhombencephalon divided into metencephalon and the Myelencephalon. But this telencephalon forms cerebral hemispheres and basal ganglia hemisphere as a cortex and basal ganglia, and the cavity is the lateral ventricle, while as diencephalon forms thalamus, hypothalamus and retina, and the cavity is third ventricles. So mesencephalon is the midbrain, stays midbrain. Cavity is the cerebral aqueduct. Rhombencephalon or hindbrain that already divided into metencephalon and myelencephalon. Metencephalon forms pons and cerebellum, and the cavity is upper part of fourth ventricles, while myelencephalon form medulla oblongata, and the cavity is lower part of fourth ventricles. This trick to remember is that to order is, of course, telencephalon is the first part, diencephalon second part, okay, and the rest are alphabetically. Mesencephalon, metencephalon and myelencephalon. So here is the important clinical correlations here is that these derivatives helps you identify where congenital defect occurs or part of brain are affected in pathology. Especially in hydrocephalus due to aqueductal stenosis, if the aqueductal stenosis affect the cerebral aqueduct associated with mesencephalon. Chiari malformation involves herniation of tonsil, the loss of encephalon structure. That means cerebral sphere and basal ganglia. So let's break down what nervous system come from which embryologic origin. Neuroepithelial cells of neural tube from structure of the CNS. Neuroepithelial neural tube. This from the structure of CNS includes CNS neuron, CNS glial cells like astrocytes, oligodendrocytes, ependymal cells, and from the neural crest cells, neural crest cell form the PNS and other tissue. PNS neuron, very dorsal root ganglia, autonomic ganglia that consists sympathetic, parasympathetic, and enteric. PNS glial cells, that is Schwann cells, satellite cells, adrenal medulla, and from from the mesoderm gives rise to microglia, which is the CNS immune cells like macrophages. Remember this: the mnemonic is neural tubes, neural PNS plus extra, and mesoderm is microglia. So there is this review in a review table of vesicles, Telencephalon. In a Telencephalon, cerebral hemisphere, basal, and T is the lateral ventricle. Diencephalon form thalamus and retina. Third ventricle cavity. Mesencephalon, midbrain. The cavity is cerebral aqueduct. Metencephalon gives and cerebellum. The cavity upper ventricles, and Myelencephalon gives medulla oblongata, gives and cavity of lower fourth. These are the very important the topic for from the neurology. You should remember this. Thank you very much.